My name is Hasegawa of Mytech Corporation. My name is Takeshi Kinjo from the University of the Ryukyus. Assistant Professor Takeshi Kinjo of the University of the Ryukyus and the Kobe Research Institute of Mytech have succeeded in developing the world's first virus visualization test method, the CV test, which can detect the novel coronavirus in about 2 minutes, making it the fastest in the world. Our CV test has 4 main features. First, let's talk about the visualization technology. CV test is an abbreviation for Coronavirus Visualization Test, which is a test method that can visualize and detect coronaviruses. The image on the right is the biochip for the CV test. When a specimen is dropped onto the round metal part of this biochip and measured with a fluorescence microscope, light is detected when a virus is present, as shown in the image below. The higher the number of lights, the more viruses are present in the specimen. By counting the number of lights, it is possible to determine the efficacy of treatment by monitoring and to determine the risk of severe disease in patients with COVID-19. In addition, when COVID-19 patients are to be discharged from the hospital, the CV test can be used for verifying that they are not infectious anymore. Next is the highly sensitive detection by saliva. The sensitivity of PCR with saliva is lower than that with nasal swabs. In addition, the antigen test cannot detect viruses of less than 100 copies with high sensitivity. The CV test, however, has been successful in detecting viruses of several copies using saliva samples. Then, there is the speed and safety of the test. The entire testing process takes only about 2 minutes, which is the fastest detection time in the world. Thus, it is possible to establish a strong quarantine system at airports and other situations where mass testing capacity is required. In such cases, the mass testing system described below is used, but in cases where urgency is required, a small device can be used to perform the inspection on the spot instead. In addition, saliva has a lower risk of infection during specimen collection than nasal wipes, and at the same time is inactivated with ethanol as the specimen is collected, thus preventing secondary infection among healthcare workers. The CV test can also be used for various infection diseases. Since the test method is based on the principle of antigen-antibody reaction, by changing the antibodies used, it can be applied by both known and unknown viruses such as novel influenza, MERS, SARS, and Ebola virus, as well as mutated viruses. Next are the results of clinical trials. Assistant Professor Takeshi Kinjo of the University of the Ryukyus will explain the details of the clinical trials and the results. I am Takeshi Kinjo. First of all, I will introduce the current diagnostics for COVID-19 and then explain about the result of clinical evaluation of the CV test. This table summarizes principles, advantages and disadvantages for the 3 tests for COVID-19, which are nucleic acid amplification tests, NAT, antigen test, and antibody detection. Currently, NAT is the preferred method for the diagnosis of current COVID-19 infections. This method amplifies target genes using spatial equipment and reagents. The merit is in its high sensitivity, but NAT needs more time, usually 1 to 4 hours, and needs skilled laboratory technicians, and the cost is usually expensive. Secondly, I will explain about the antigen test. This method detects a specific protein of the virus. You can get the result in 15 to 30 minutes, without special equipment and reagents, but the sensitivity is low compared to NAT, and you can't test saliva with this method. Lastly, antibody test detects antibodies in the blood. Antibodies are not produced sufficiently in the early phase of infection. Therefore, antigen test cannot be used for the diagnosis of current infection. As explained above, current diagnostics for COVID-19 have several limitations. To inhibit COVID-19 pandemic promptly, distribution of useful diagnostics is important, and development of novel, rapid, easy, and high-output methodology is needed. From this viewpoint, I think the CV test is very promising. In this slide, I will show you the result of a clinical evaluation of the CV test. First of all, we examine the accuracy of CV test using stored clinical specimens. In this first evaluation, 50 COVID-19 patients and 20 non-COVID-19 patients were included. We tested nasal swabs from these patients with both CV and PCR tests. Clinical samples for the CV tests were mixed with equal volume of ethanol. Therefore, the final concentration became 50%. CV test was very easy and took only 2 minutes to get the result. On the other hand, the procedure for PCR was a little bit complicated. You had to exclude nucleotides from the samples, then mix them with PCR reagents, and finally to insert them into the PCR equipment. It took about 4 hours to get the result. Before I explain our result, I will explain how to interpret the AUC value of the ROC curve. Software for statistical analysis draws the ROC curve, and as the area under the curve, or so-called AUC, is larger, the tested method is more reliable. Generally, an AUC of 0.7 to 0.8 means acceptable. An AUC of 0.8 to 0.9 means excellent. An AUC of more than 0.9 means outstanding. The AUC of the CV test in the first evaluation was 0.98, which means outstanding. The positive and negative agreement rate were 94% and 100% respectively. In the second evaluation, we tested with freshly collected nasal swabs and saliva from 55 COVID-19 patients and 50 non-COVID-19 patients. The method was similar to the previous evaluation, but samples used for the CV test in the second evaluation were mixed with double volume of ethanol. Thus, the concentration of the original patient's samples were thinner. The AUC of nasal swabs and saliva were 0.84 and 0.87 respectively, which means excellent. 2-by-2 table gave the same result for both nasal swab and saliva by chance, and the positive and negative agreement rate were 72% and 96% respectively. When focusing on the lower copy samples, the CV test showed positive in 11 samples out of 14, which viral copies were less than 100. Now, let me explain the latest technology that makes CV testing possible, quantum crystals. Quantum crystals are a novel substance generated by three-dimensional self-assembly that has been successfully developed by Mytech. When antibodies and quantum crystals are mixed and dropped onto the biochip substrate, the antibody solidification process, which normally takes more than 12 hours, can be completed in just 1 minute. This achieves a rapid detection time of about 2 minutes for the entire process. It is also very important to distribute the antibody solidification throughout the entire process. For example, if there is any bias in the fluorescence, accurate quantification will not be possible. As you can see in the image, if the quantum crystals and antibodies are evenly dispersed, the fluorescence that shines on them will also be dispersed, allowing for accurate counting and quantitative detection of the virus. Furthermore, since quantum crystals are plasmoic materials, they have the effect of enhancing fluorescence, making it possible to visualize viruses with high sensitivity. These features allow for rapid detection, quantitative testing, and high sensitivity. In addition, when COVID-19 subsides and in the future eventually new viruses become prevalent, the quantum crystal can solidify any antibody because it is a metal complex. Therefore, it is possible to test for known viruses such as Ebola, MERS, SARS, and novel influenza, as well as unknown and mutated viruses by changing the antibody to be used. The quantum crystal technology has already been granted substance patents in many countries around the world. A patent application for the CV test has also been filed under the US Prioritized Examination Track 1 system. Here is the virus detection procedure. All the specimens that contain a certain amount of virus can be tested. Let's use saliva as an example. First, the specimen is collected and, at the same time, inactivated by mixing saliva and ethanol. Next, in step 1, antibodies and quantum crystal reagents are mixed and dropped onto the biochip, and the antibodies are solidified in 1 minute. Then, in step 2, the collected sample is mixed with the labeled antibody. Here, if there is an antigen, for example a virus, the antigen and the labeled antibody will bind first. After the antigen is added to the sample, the antigen and the antibody are combined in a 1-minute antigen-antibody reaction. Once everything is bound, the labeled antibody remains even after washing, and the test is completed in about 2 minutes by detecting the fluorescent light with the testing device. In summary, it is a very fast and simple test method. Drop a solution, 1 minute, drop another solution, 1 minute, wash and measure. Now let's talk about the mass testing system, which is used to process a large amount of the protocols mentioned earlier. The mass testing system consists of a detection machine and a fully automated pre-processing system. The detection machine is made by Olympus, a world-renowned company. The schematic diagram on the right shows the system that automates the pre-processing, steps 1 and 2, to feed the biochips. By using this system, we can maximize our testing capacity. The system can process 4 specimens per minute, 240 specimens per hour, or 5,760 specimens in 24 hours per day. This makes it possible to easily detect infected people at airports, large-scale events, and other places where an unspecified number of people visit or pass through in a short period of time. In addition, Nosocomial infection prevention at medical facilities and regular testing at companies will help prevent corporate infection and prevent loss of productivity. I will now explain our future plans. University of the Ryukyus and Mytech plan to optimize the CV tests to further improve accuracy. We plan to conduct clinical trials on the application of this test to the pathogenesis of infections and the determination of therapeutical efficacy based on the changes in antigen levels. Mytech will accelerate the mass production of the CV test system and will promote the licensing of the technology and the distribution of the system to countries and regions where the infection is spreading.